So, hello everyone. So, hello everyone. Am I audible to all of you and my slides are visible?
Yes, ma'am.
Okay, so we will start the today's unit because this is very important. This is all experimental based, research based. So, we will study it in a very basic way because you might be not aware about the techniques and what is the different machines used for all those things. So, we will study this in a bit more detail so that you will be able to understand each and every thing. So, let me do the slide play. Okay. So, in this, firstly, we will understand the next generation sequencing. Why it is known as next? What does this mean by the next? And what is its role in the microbiome analysis? And what is the role of bioinformatics, or you can say computational biology, in microbiome research, or how it can lead to the advancement in the gut-based microbiology? And apart from the bioinformatics, nowadays we also have AI and AIML machine learning. So we will see that how this artificial intelligence and machine learning helps to develop, or it can also lead to the advancement in the microbiome based diagnostic. Basically, it has rapid the rate of research going on just because of the database we have. We can just look our query sequence in that. We don't have to wait for the people to upload their data in that. So already we are having different kind of database related to each and every type of transcript data. You can say DNA data, RNA data, protein data, gene mutation data, or everything is available at NCBI. Now, what are the ethical and regulatory aspects in microbiome therapy? So we will try to cover these 2 points in today's video. So let's start it. So this is the, you can say, basic compilation or basic summary of what we do by the help of NGS in microbiome analysis. So mainly, we study the composition, like the gut microbiome is rich in different kind of microorganisms. So which type of microorganism, what is the structure of that microorganism, and the function of the microbial communities in the gut. So with the help of next generation sequencing, we will be able to know about the study of the composition. So let me take a black screen. Hmm. Suppose if this is a person, this is a person A. So this is an healthy person. So if you will analyze the gut microbiome of this person, so you will analyze that. So for analysis, what are the techniques? What are the different kind of techniques we are having in molecular biology so that we can analyze the uh, different composition of the microorganisms residing in the gut of this person A. So we will go for the 16S rRNA. If you remember, I have taught this in my very first module, the 16S rRNA sequencing, because this act as a signature nucleotide, or you can say this is the sequence which is specific for each and every individual. So you will go for the 16S rRNA sequencing of all the microorganism, like whatever we have studied, that trillions of microorganisms are there in our gut. So what you will do, you will extract out all those microorganisms. First, the first step will be microorganism isolation. You will isolate the microorganism from the gut of this person A. And after the isolation, what you will do? You will isolate the genomic DNA. GDNA means genomic DNA, because a bacteria will have its genomic DNA. So you will do what? Extraction of the genomic DNA. So in molecular biology, the basic step, whatever you want to do, whether you want to check the expression of any gene, whether you want to check that this genome, uh, whether the bacteria is having this gene in its genome or not, because we don't know. Whenever we perform the gene expression analysis is different thing, and the gene analysis is different thing. Expression means already the gene is present. We want to know that whether it is expressing at a higher rate or a lower rate. So gene expression is a different term, and we are finding that whether a particular X gene is present in its genome or not. So for that, the foremost and the basic first step is extraction of the gDNA. You will do the extraction of the gDNA by removing all the carbohydrate, protein from the cell, and then you will directly extract out the gDNA. And after that, you will amplify. Amplification means you will do PCR of particularly this 16S rRNA gene. So for amplification, you will need primers, and for primers, you are already having universal primer that will definitely bind to the 16S rRNA sequence of almost 99% of bacteria. So you will amplify that, and after that you will go for sequencing. So this is the basic step. It means you have to isolate the microorganism from the gut of this uh, person A, and then you will isolate the genomic DNA, and then you will amplify it. And after amplification of this particular gene, because you have already amplified the signature gene, now you need to know what are the sequence of its gene in order to determine which organism is that. So you will go for sequencing. Now if you want to go for any protein analysis, that whether this gene is expressing at a higher rate or a lower rate, so after this gDNA formation, you go for the from gDNA to cDNA. You prepare the cDNA, because every expression, every protein related expression is checked from this cDNA. As you all know, cDNA will is a transcript which will allow you to know whether this gene is expressing at a higher rate, lower rate, as compared to the housekeeping gene. Because always we know housekeeping genes are the constitutive gene which are constantly in on position, like the gene required for the survival, the gene required for the protein synthesis. So all those important genes which are required for the normal physiology of a bacteria will be always expressed constitutively. It means it will be a housekeeping gene. So it will be always in a on mode. So comparing it with the housekeeping gene, we say that twofold increase, twofold decrease. So this is known as the gene expression wala part. Now, for the gene expression, you will go for gDNA to cDNA. So this is the extra part which I wanted to let you know. So we have to stick on this one. So this whole part, like from microorganism isolation, gDNA isolation to sequencing, this part is a crucial part which will allow you to know that which microorganism is there in the gut of the person A. So likewise, you will rectify that it has Bacillus subtilis, it has Clostridium, it has Bifidobacterium, it has this, it is that. So you will identify, and after that you will group as per the phylum, because we have phylum of bacteria. So you will group that this is Bifidobacteria, this is Bacteroidetes, this is Vericomicrobia. So you will identify that. So you have to go for this process. So this process is a very before the discovery of this NGS, this process is very time taking and very exhaustive, tedious, and it is not possible for all the organisms to be sequenced. So for that, people have developed, researchers have developed next generation sequencing, because every time the research, if we observe the research chronologically, so you will see this way, basic to advanced. Because obviously people will develop something very easy very fast, and after having some sort of restrictions, you can say, limitations, you can say. So that thing is modified little bit, and then advanced things are into the market. So this is when firstly we have Sanger sequencing. Sanger sequencing. So this is the overall basic overview I wanted to let you know. Now see, the before going on to directly next generation sequencing, you should know about, I want clear page. So I have taken this slide. Okay, so we have space here, we can understand here. So firstly, Sanger sequencing was done. Sanger sequencing is also known as chain termination. Chain termination method, or chemically it is known as dideoxy method, because in this the 2' carbon lacks the oxygen. So you can say, sorry, the 3 dash carbon. So in this, you all know the role of DNA polymerase. What do the DNA polymerase do? So it elongates the growing chain to form the new daughter strand. So what does it require? It required 3' OH plus 5' phosphate. So it makes a phosphodiester bond in between them. Phosphodiester bond is made. So if this 3' OH is not available, then this DNA polymerase will no longer be able to extend that growing chain, and then the DNA replication will be terminated. So this method is known as chain termination. Whenever the growing chain will incorporate some dideoxynucleotide, the DNA polymerase will no longer be able to synthesize that chain, and that accordingly some stretches will be there. So you can analyze this fragment, this fragment, this fragment. So this is known as Sanger sequencing. Now since 2 to 3 decades, NGS is introduced. You can say the advancement is since a decade. So NGS is applied to this conventional Sanger sequencing. So everything was done manually in Sanger sequencing. You have to add the DNTPs, you have to add the dideoxy DNTPs, you have to add the primers, and then the sequencing was performed. But can you tell me that all these things are possible if you have 1 bacteria, if you have 2 bacteria, let's say 10 bacteria, but this is not at all feasible if you are having more than 1,000 bacteria, or more than 100 also, I can say. This is not possible for a person alone to do all the sequencing of more than 1,000 millions of things. So for that reason, everything is, this Sanger sequencing is automated. Means no more manual thing, no more labor requirement. Only a single person can do the sequencing of more than 1 million genomes overnight. So all these things, you can say, thanks to next generation sequencing, or the sequencers developed by different institute and different companies, you can say. So all these things are very handy, and they take almost you can say 1 to 3 hours is 3 hours are enough for the sequencing to be done. So this is the principle behind the next generation sequencing. Now I will come point wise. So for why we use this next generation sequencing? For the study the composition, that what our gut microorganisms are from different phylum. So we want to know that each person is having different kind of microbial diversity in their gut. What is its structure? So how structure can be determined? Because you can, through sequencing, you will get the nucleotide sequence. So through nucleotide, you can convert it into a codons, the protein coding genes. So through that, you can predict the 3D structure of a any gene, any molecule. And there, how you can know the function of the microbial communities in the gut. Now, main sample type will be obviously the fecal sample, because it act as a source for isolating gut microbiota. So whenever you will complain about having some gastrointestinal disease, then you will be able to diagnose your fecal sample by your pathologist. Now, what your pathologist will do, whenever he or she will harvest fecal samples, then he or she will isolate your DNA extraction will be done. This genomic DNA. Genomic DNA will be extracted out, its purity is checked, because 1.8 is required for the DNA purity. So for PCR, we also do 1.6 is also acceptable. But if you for the first time, or let's say if you want to go for the uh, sequencing, then there are certain formalities or the criteria you need to fulfill before giving your sample to the sequencing facility. They require a clear band in the agarose gel, a sharp band, not diffusible, saturated. A sharp band is required, and a concentration of 1.8 is required. 1.8 is a denotes that your isolated DNA is pure. 260 by 280 nanometer we take the absorbance at 260 by 280 nanometer. So if it is 1.7, then also good. If it is 1.8, this is excellent, that your DNA is free from any kind of impurity, no carbohydrate, no protein, no RNA impurity, nothing. So this is the foremost step. So they check the purity, because this is also important, because if you are having an impure DNA, then you are using that as a template for PCR, then your PCR bands will always be like it will not be acceptable. So always try to have whenever you are taking DNA as a source or as a sample, as a template, then it should be of pure concentration 1.8, or else not less than or more than 1.7 or 1.8. So whenever you will go down, something will be happened. If you are going up, if your ratio is less than 1.8, you are having protein contamination. If your ratio is more than 2.0, you are having RNA contamination. So that is not a good thing to go in the molecular biology, because you need the proper ratio of DNA. And after that, these are the techniques which you can go for. So first and the foremost is always in microbiology, you can say the 16S rRNA is the very basic and the first step, because we have to identify the bacteria taxonomic profiling. So you have to identify that bacteria, its genus, its species, and if you identify the genus and species, you will be able to identify the taxonomic group, the taxa. Now after that, you can go for metagenomics. Metagenomics means whole genome sequencing of all microbes. Now see, uh, people will ask you, like we have communicated one paper which is accepted, fortunately. So we have done the 16S rRNA of a bacteria which was a Bacillus cereus MHS strain. We have done only the 16S rRNA because we wanted to identify the bacteria and nothing else we wanted to do. We only have to identify that is this a Bacillus cereus or not. So for that, 16S rRNA is good enough. You don't need to do anything other than that. So we have done that. You will see more than 99% of the paper that people do 16S rRNA, and this is more than enough if you want to identify that particular organism. But one of the reviewer raised the question that whether you have gone for the whole genome sequencing, WGS, whole genome sequencing. So see, this is the genome of the bacteria. So we have only done the sequencing of the required gene, that is the 16S rRNA gene, which is known as the signature gene, or you can say this is for the identification of that organism. So we have already done it, but the reviewer said they actually recommended us that you can go for the whole genome sequencing. So if you are doing the 16S rRNA, then you can say 99.99% surety is there that if you have identified that this is Bacillus cereus or whatever be the species or bacteria, so you are only 99.99% sure, which is acceptable, nothing wrong in it. But if you are doing the whole genome sequencing, it means not only just this part, 16S rRNA wala part, but the whole genome of the bacteria. So you can say you are 100% sure that this is the Bacillus cereus. But people do not follow this whole genome sequencing. The reason are it is costly. Because obviously, if you are sequencing for this much 1500 base pair, and if you are going for this 4.6 into 10 to the power 6, 7 base pairs, so obviously this will raise the cost, because it depends on the length of the gene which you want to go for the sequencing. So this is costly, around 20K, 25K. Also, it depends from different institute to institute, but basically you will need 20K for a whole genome sequencing. And this is very tedious task. You have to go for the whole stretch of the genome. So this is time taking also. So only 16S rRNA is fine. So we have just written a explanation, like justification that it is not required. We have done the 16S rRNA sequencing and we have gone for the phylogenetic tree analysis. So that is more than enough. So he agreed and he accepted the manuscript. So these are the things going on in research. So if you are working on any microorganism, just go for the 16S rRNA if you are working on prokaryotes, and for eukaryotes 18S rRNA is fine. You don't need to go for the zabardasti ka whole genome sequencing, because this doesn't make any sense. We are increasing the cost of our research, and no one will ask you to give the whole genome sequence of that particular organism you are working on. They will only ask that provide us the unique 16S rRNA sequencing and the phylogenetic tree. So metagenomics, you can also do whole genome sequencing of all microorganisms. Metatranscriptomics. Transcriptomics means the study of the RNA transcripts that is related to protein. If you are analyzing the RNA, then you can go for directly the metaproteomics, that how many different kind of proteins are manufactured by the microorganism and at what level. So for this, you will be doing work with cDNA. You will isolate the gDNA, you will convert it into cDNA, then you will use that cDNA as a template for PCR to analyze that which gene is expressed more, which gene is expressed less. And apart from this, metabolomics. Metabolomics means, as the name suggests, study of the metabolites, that this microorganism synthesizes this particular genotoxin, this particular endotoxin. So for that, you will go for metabolomics. So for all these things, you need to isolate the, firstly, DNA, or you can say the genomic DNA of the bacteria. So these are the common techniques which people use nowadays. And on top of that, 16S rRNA is the preferred one because it is hassle free, it is cheap, it is quick, it is reliable, you can say. So all those things are associated with this. Now there are so many sequencing platforms like Illumina sequencers are there, Roche sequencers are there, ROCHE, Roche 454. So there are so many different kind of variant associated with this Illumina. Now we have Oxford Nanopore. We have like we have what to say? I forgot the name. Uh, Gen Biosys. I think we have the one more variant related to this name. I will confirm it. So these are some of the manufacturers of sequencer. So in many of the institute, you will see the sequencer manufactured by Illumina, by Roche 454, by Oxford Nanopore. So this is the latest one. You only need only 1 to 3 ml for the whole sequencing to be done. PacBio is also there. So there are different options of sequencers. We have, let me show you this card. So I can show it with my some previous PPT. DNA sequencing. So DNA sequencing was one of my topic of my PhD seminar. So I have prepared this in a very detail. Mm. So see, let me just, this is the whole genome sequencing process. See, the bacterial culture is, after the bacterial culture, we extract the DNA, and after that, we digest it. And after the digestion, we go for the sequencing, and then we sequence it, and then we check the similarity with the existing one. So, what are the recent advances in DNA sequencing? So, we have next generation sequencing. See, these are the company 454 Roche and Applied Biosystem, Life Sciences. These are the variants. Like we have so many generations. Am I able to use now? So in this, whenever you read that next generation, NGS, second generation, why these things are added to the sequencing? Because these are the latest development, or you can say advancement in the conventional Sanger sequencing we are having. So you have to remember, you cannot forget the Sanger sequencing, because this is the basic principle. All the recent or the latest sequencing technology rely on this Sanger sequencing only. The principle is Sanger sequencing, but instead of the manual thing, it is automated. Like no labor is required, no more manual thing. So you cannot say that Sanger sequencing is not reliable. People will ask you which sequencing you use. So we always use Sanger sequencing, but the methods are different. You can use Illumina, you can use Roche 454, you can use Pyro sequencing, you can use Ion Torrent, which is the latest one. So these are the sequences you can see. So this is Ion Proton, this is Ion PGM, Illumina. This looks like most of the places have Illumina. We also have Illumina in our ICER Kolkata. And we have also one more variant, and this is Roche 454. So these are the sequencer. This looks like a more or less like a PCR machine, because PCR also looks like this only. So they are compact, they do not require that much of space. And these are the, this is an Illumina sequencer. You can see these are the sample holder. This is a screen where you put all the information about the run of your PCR cycle, because obviously you have to do the PCR before the sequencing. So these are the methods. I will not elaborate this. I only wanted to, this is the Ion Torrent sequencer. So this is a tube like where you can put your tube in which your samples will be there. So this is an approximate cost also. Like if you have this much of read length, like 450 to 900 base read. So what will be the cost to $10? Illumina sequencing will have $0.07. So this is, people use most of the time Illumina sequencing because this is cheaper. And this one is of very highly expensive. Oxford Nanopore is the latest one. See, this is a very handy one. You can also keep it in your pocket, and few drops you will put into the sample holder, and then you will be good to go. You will be provided sample. So this is one of the variant which is latest, but mostly this variant is used, not this one. This one, Illumina sequencer. Most of the places you will see Illumina sequencer. Okay, now back to our PPT. Okay, so after that, the sequencing platform we have already studied. We have different bioinformatics tool. Because why bioinformatic tools are required? Because the sequencing will generate the sequencing data, the sequence data. So what you will do without bioinformatics to that data, that huge amount of data, that's the lengthy stretch of sequences you will get. So let me show you sequences, how the sequences looks like. So I will show my sequence. So whenever your sequencing result will come, I can show any of the result. Na? So, okay. Okay. So your sequencing facility will provide you two files. This is for my two bacteria. So for one organism, you will be provided two files. So you can see here FP. FP is forward primer and RP is reverse primer. So always whenever you are going for this sequencing, your sequencing facility will provide you two files for each organism. So I was having two organisms, so four files were there. So this is the forward primer. So whenever you will open it, firstly you will need that software to read that file, because they provide the file in .ab format. So that is not readable by any of the app. So for that, some sequence reader, like you can go for BioEdit. There are so many different kind of software which you can easily install in your laptop, so that you can read the sequencing result. So I have installed the BioEdit. See, this is AB1 file. So for that, you need a particular reader, faster reader. Okay, so this is okay. So that both those files were having some problem. So this is the sequencing result you got. So you can see these are stretches of the nucleotide, that starting from C, G, 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 G to up to the last. So this is known as the chromatogram. You have received this peak wala thing is known as chromatogram. Like the thymine is tagged with red color. Like whenever you're sequencing, individuals will do the sequencing, they tag each and every nitrogenous bases with a particular color, so that it will be represented easily. Like red denotes what? Thymine. Blue denotes what? Cytosine. And G is guanine is in black color. Now adenine is in green color. So these four colors are used for the sequencing. So this is the advancement in the Sanger sequencing. In conventional Sanger sequencing, no such fluorescent dyes were there. People do manually. So but in next automated sequencing, after the automated sequencing, and from that, each and every nucleotide is labeled, fluorescently labeled, so that we can be able to know by the signal. So you can see only four types of peak are there. So you can scroll and see. So if you are getting this sharp peak, it means your sequencing results are good. You can rely on that. So if you are getting some noise, like see, here it is noise, so this is not a problem, because whenever you will go for sequencing, whenever you will do PCR, so the primer will bind to your template. So up to some stretch, like let's say 19 base pair, you will have some noise, and after that you will get sharp peak. So this is the you can say technical thing which is related to your sequencing. So people will do what? We can cleave this wala part, because we know that primer is getting amplified. Primer is amplifying our target sequence. So for that, it takes some time, and after that the peaks come. So we can chop off this part from the beginning also and from the end also. So this is the processing part after getting the sequence. So you can see there are so many sharp sequences. This is a, you can say, good sequencing result. It means I have used the DNA template, which is pure, which is 1.8, and all the primers were of good quality, no contamination, nothing. So you will get a peak like this. So as I told, from the beginning and from the end, you will get noisy peak. So this is nothing but the normal technical thing of your sequencing. So this is how it looks like. So you can say that is known as the chromatogram, where peaks are visible. Now this is the exact sequence if you want to see. So see, sequences are there. You can scroll and see all the sequences. So this is the only, you can say 390, 1, 2, 3, 4, 5, 6. 396 base pairs are there. So this is forward primer. It means this sequencing is done by the forward primer. We in PCR we use two primers. So to confirm, let me draw it and no. So whenever we go for sequencing, laptop is again hanged. So whenever we go for the sequencing part, we are having forward primer and we are having reverse primer. So it is recommended to do the sequencing with both the primers. You cannot rely on to the first one because we have to make a contig. Contig means you have heard this many a times while reading the sequencing. Contig means nothing but the stretch of sequencing result you have obtained through the forward primer and the reverse primer. We add both the sequence. Contig is also known as consensus sequence. So these are all the technical things associated with the sequencing part. So for the major, you can say for the reliability purpose, we go for both the forward primer and the reverse primer. We cannot go only for the forward and only for the reverse, because whenever you are going from this way, the sequence will be A, T, G, C, T, C, and whatever. And if you are going from this way, it should be like C, T, C, G, T, A, and nothing else. So this will be 100% reliable that yes, the sequencing result is uh, correct and we can rely on it. So for this reason, we go for forward primer and reverse primer, both sequencing. So people just for the saving the research cost, they go for only forward primer and reverse primer, but that is not acceptable. You need to make a contig, and contig can be only made if you go for the sequencing from the forward as well as reverse primer. So now this one. So there are different kind of bioinformatics tool, and you can use these tool for the analysis of whatever sequencing data you have got. So for the basic thing, you can refer NCBI. You can see the homology, that whether this is existing in our literature or not. And we have different kind of databases also. And uh, the major application is through that you can say disease biomarkers. Like you can say if your gut is rich in this bacteria, you may have chances of IBD, you may have chances of TD, T2DM, that is type 2 diabetes mellitus. And accordingly you can design personalized nutrition, that you can eat fiber rich diet, you can eat protein rich diet, you can eat this, that. Antibiotic resistance is the major application. Like if people are developing antibiotic resistance, if bacteria is developing antibiotic resistance, this is a bad thing. Like no antibiotic will be act on their cell. So how we can do that antibiotic resistant tracking, gut brain axis study. So everything is possible if you are incorporating the next generation sequencing in your gut microbiome wala thing. Now, challenges are sample variability, because we have a lot of sample. If you are isolating the gut microbiota, so you can recall that our gut is full of different kind of microorganisms. So how that microorganisms will be handled? So it is very difficult to maintain the sterile condition. Like no such cross contamination should be there. Like we are having millions of bacteria, A, B, C, D, 1, 2, 3, 4, likewise. So how to maintain the pure culture of A, pure culture of B, C? So microbiology is easy, and at the same time it is very very risky thing. Risky means your sample will get contaminated, so you have to go for the pure culture. You have to go for the again sequencing wala part to confirm that this is the required strain or not. So in microbiology you can do a lot of research, but the basic should be up to the mark. Like top notch research is required, but you have to take care of the basic organisms you are using and how you are routinely maintaining that culture. So And that is an important thing. So these are the things, like I just talk about the contamination thing. So contamination is one of the, you can say, the major antagonist of the microbiology. Like whenever you are handling two cultures at a time, then it cross contamination will be there. So whenever we work with two organisms in the same lab, or you can say more than, obviously everybody will work at with some different microorganism. So the hood we use or the laminar we use, we always go for the uh, partition wala time. Like if I am working on Bacillus cereus, so I will do the work, and after that I will I will maintain the sterility of the whole hood chamber. Like I will go for the UV sterilization for 40 minutes at least, so that no more bacillus will be there. Because cross contamination is the very problematic thing in microbiology. So people don't take care of their culture. Cross contamination happens. So this is the case whenever you send your sample. If no in-house sequencing facilities are there, if you send your sample for 16S rRNA, then that sequencing facility will send you a mail that your samples are impure. Like if you are having mixed culture, so many a times all these things are sent by the sequencing facility. So these are the major hurdles, you can say, major stoppage, major challenges in the way of this next generation sequencing. And obviously data complexity, because we are having data of millions of bacteria, so it is a very difficult thing to handle. So I am right now working on to 6 bacteria, so this is a difficult thing to handle each and everything of that 6 bacteria. So I have now optimized that I will work only on one, the hyper producer, the hyper remediating strain. So but at the initial level you have so many strains to work with, so you have, it is very difficult and very confusing, because you have to label the files properly. So all these things are a mess if you are not handling anything properly. So all these things are related to the actual research part, actual, if you are having a good experience in all those basic microbiology wala part, so everything will be fit and fine. But you have to be very you have to take precautions whenever you are working with more than one microorganism. Now from this, you need to focus on the 16S rRNA gene, which is a highly conserved gene used for bacterial identification. I have explained this point so many times. And operational taxonomic unit that are known as the OTU. So OTU are regarded for those who have similar sequences used to classify bacteria. Suppose OTU, in OTU1 we have A, B, C, D. So what you can say, the bacteria A, B, C, D have similarity in their sequences. Therefore, OTU1. Now, OTU2 have some E, F, G, H, likewise. So, these are the group of similar sequences used to classify a bacteria. Amplicon sequences means after the PCR sequences you are having. So PCR based targeting a specific region, that is 16S. So this is known as amplicon. Amplicon is nothing but the PCR amplified fragment, that is known as amplicon. Shotgun metagenomics. So random sequencing of all microbial DNA in a sample, that is known as metagenomics. If you want to scan any of the gene in any of the bacteria, you will go for random sequencing. Like suppose gene X, you want to see that whether it is present in bacteria A, B, C, D, E, F, G, H. So you will randomly choose any of the gene like gene X, and you will use the primer of this gene X, like forward primer, FP and RP, and then you will do the PCR. So whenever you will get the band, you can say, suppose in A you got band, in B you got band, and in all of the and rest of these you do not get any band. So you can say only A and B have this gene X in their genome. So this is known as metagenomics. Now, how we can incorporate bioinformatics and microbiome research? So the purpose of incorporating bioinformatics is this part, the data complexity. How we gonna handle those huge amount of data generated by the sequencing, because our gut is already having millions of bacteria. So for that, we need bioinformatics so that it can be grouped. The bacteria can be grouped as per the similarity in the sequences. So accordingly, we can group A, B, C, D into phylum 1 as per the similarity, E, F, G, H into some other phylum. So we can just categorize it for the sake of our convenience. So the purpose is to analyze large scale sequencing data from microbial communities, especially gut microbiota. Now the sample source is always the fecal sample. So fecal sample act as a source to isolate the whatever gut microbiome we are having. So apart from this fecal sample, you can also use mucosal biopsies, saliva, skin swab. So from all of these things, you can isolate the bacteria because bacteria is everywhere. Microorganisms are everywhere. So what are the omics techniques we are having? So we are having, you can quickly recall the metagenomics, the metatranscriptomics, the metabolomics. So every omic we are having. These are the major kind of omics in research. So core bioinformatics steps are quality check, trimming and filtering, assembly, taxonomic and functional annotation. Annotation means identifying that this belongs to this group, this belongs to this species, genus, and likewise. So firstly, we check the quality. As I showed you my result, that was not opening, so that was not of good quality because something happened to that file. So when I opened the correct one, so it has a good quality of peaks, were there sharp peaks, so you can easily identify the sequence you are getting through the sequencing result. Then you will trim and filter. Trim means I just told you that you need to trim from the beginning and from the end because noise are there. So you need to go for only the this wala part. So this is known as trimming and filtering. Assembly means suppose you have more than 100 sequence. So you do this wala. See, alignment wala part is known as assembly. After that, you will do BLAST, basic local alignment search tool by the use of NCBI, and then you will be able to know that which bacteria is that there. Bacillus is there, bifidobacteria, lactic acid bacteria, whatever. So key tools are fast QC, quality check. For quality check, we use fast QC, quality control. And also we for trimming, like I use BioEdit because this is an easy software, easy interface for a software. I never use any software that is very heavily loaded because many a times whenever it runs, it will hang. It will hang the laptop and you have to do again and again. So always try to have a free software, not so heavy one, not so sophisticated one. BioEdit is a very acceptable and majority of people use BioEdit. So there are some tool for functional annotation. You can refer to databases, and these are some of the database like Green Genes or RDP, NCBI reference sequences, KEGG metabolomics. For metabolic pathway, we have KEGG pathway. So all these are different softwares. So for diversity analysis, alpha diversity, these are some formula if you are aware about the ecological thing. So we have Shannon's index, we have Simpson's index. So all those things are there. Now for visualization, for statistic, for different functional prediction tool, we have different kind of softwares assigned in bioinformatics. So for this reason, we go for the bioinformatics analysis, and it becomes easy for us to go and do a lot of thing with whatever sequencing data we have received. So this is again the same thing. So see, I have always provided the difference between microbiome and microbiota. So microbiome means the whole genetic content, and microbiota means the community of microorganisms. When I say microbiome, so I am referring to the genomes of all the microorganisms residing in the gut. But if I am saying microbiota, I am referring to the whole some cell like microorganisms. So this is the same thing. So you can see So why it is known as next generation? Next generation sequencing, also known as massively parallel sequencing, is a method of simultaneous sequencing of millions of fragments at a time without previous sequence knowledge. You don't need anything. You only need to what we give to the the people who do the sequencing. So we provide our gene which has to be sequenced, and along with the gene we also have to provide the forward primer and the reverse primer, because obviously we have amplified that particular gene from the whole genome. So we need to provide the forward primer and reverse primer so that they will use the same primer to do the PCR again, and after that they will do the sequencing wala part. So the main utility of NGS in microbiology is to replace conventional characterization of pathogen by morphology, staining properties, metabolic criteria with a genomic definition of pathogen. So see, whenever we go for the isolation of microorganism, we go for the morphological characterization, the whether the bacteria looks coccus or bacillus or vibrio or something else, and after that we go for the texture, color, elevation, that is known as morphological characterization of the bacteria. And through that we also do the staining, gram staining, acid fast staining, and endospore staining, you can say, and a lot more. So all these things are, after doing a lot of this biochemical test and morphological test, you write it down like this is a bacillus. But you cannot rely on to your biochemical and morphological test. Nowadays it is not at all acceptable that only you have gone to the morphological parameters and biochemical parameters. So this is replaced by simply 16S rRNA. By the help of this NGS, by the help of this NGS, you are able to identify that bacteria without going this uh, what staining wala thing and this morphological characterization, because you cannot rely on to only the staining part. So this is for the characterization of pathogens mainly. Now, next generation sequencing is a general term that describes the class of high sequencers that have been developed after chemical degradation and chain termination method. So chemical degradation was given by Maxam Gilbert sequencing and chain termination by Sanger, which is one of the widely accepted sequencing. So due to next generation sequencers, output of millions or billions of sequence read per instrument run, the process of genome sequencing has been revolutionized such that a single laboratory technician can now single-handedly produce whole genome sequence data for multiple bacterial organisms overnight. So this is the main thing which I have explained to you, that it is not possible for a single person to do millions of gut bacteria alone by a manual thing. Nobody can do that. So to replace that, we only need a single laboratory technician that can overnight do your billions of sequencing. So this is the major advantage of next generation sequencing. So this is the basic overflow, like sample preparation, library preparation, then clonal amplification. As I told, PCR is done before sequencing, then sequencing by synthesis, and then data analysis. So these are the things which takes place. So this is the difference between the Sanger sequencing and the next generation sequencing. So Sanger sequencing, which is known as the first generation, and after the advancements in this one, it is known as next generation sequencing. So the throughput was low, like it can only do the one gene per run, but it can do the whole genome per run whenever the cycle goes like this. So only itna stretch is possible for Sanger sequencing, but itna stretch is possible for NGS. So you can imagine that it can go whole genome per run and it can only do one gene per run. The speed is very slow. We have to wait for so many days to get the sequencing result, and this is fast. The cost is high per base. This is low per base. Long read length is very long. This can read a short length, because whenever we do sequencing we get stretch like this. So for Sanger sequencing, at least itna stretch is readable. if you get any this stretch, it is not readable by the Sanger sequencing. But in NGS, even this much of stretch can also be easily read. Scalability is obviously limited, low, because it is very costly. It is slower. It can only have one gene per run. So this is modified into next generation sequencing. So this is massively parallel. Massively parallel means you can do millions of sequence at a time. So why next generation? Because NGS represents a very advanced latest technique. It can perform millions of reads simultaneously. You can do whole genome, transcriptome, metagenomics, metabolomics, anything you can go. So this is nothing but a revolution. So you can do a lot of things at a single time. So this is the overall workflow. Now I've also included here also. I forget to that I have also included in this PPT. So iron torrent, PacBio. So all these are variants of different sequences. So more or less, it looks similar, despite having different brands. It looks similar only. We have screens attached with the sequencer so that we can feed our input. Like we have to go for this much cycle. We have to go for this concentration of the template, and everything is on the screen. Like how much progress has been done. When it is completed, it will show you that the cycle is remaining, this much amount of time will be required to do that. So all these things are automated. So this is all for today. Now we will continue tomorrow.